Hi, I'm Marianna Bacellar-Galdino, and I'm a veterinary specialist here at Altasciences Preclinical Scranton. And I'm here today to talk about the poster entitled A Comparative Study of Ocular Anatomy in Three Different Laboratory Swine Strains. So the objective of this study was to show that there is many similarities between the eyes from three different strains of pigs for preclinical research. So the use of swines in ophthalmic research has grown considerably due to their anatomical and physiological similarities to humans, especially the retina when thinking about the eyes. So we compared eyes, size and weight of tissues from Sinclair nanopigs, Göttingen mini pigs, and Yucatan pigs. They are commonly used in toxicology and pharmaceutical research, and they are well known to be good species, especially for ocular studies. So the total of animals that we used was 49 animals. We had 32 eyes from Sinclair nanopigs, 12 eyes from Göttingen mini pigs, and 5 eyes from Yucatan pigs, with ages ranging from 1 to 30 months old. So the eyes were either dissected, 24 eyes from Sinclair nanopigs, 10 eyes from Göttingen mini pigs, and 6 eyes from Yucatan pigs, or fixed in 10% formaldehyde. So we had 40 eyes from Sinclair, 14 from Göttingen mini pigs, and 4 eyes from Yucatan pigs. Uh, for they were fixed in formalin for overnight for evaluation the next day. So the dissected eyes had all the subocular tissues collected and weighted. So aqueous, vitreous humor, cornea, iris, ciliary body, lens, trabecular meshwork, retina, choroid with RPE, and sclera. And the eyes that were fixed, they were sectioned sagittally for measurement of the anterior posterior, so axial, and transverse or horizontal global lengths, cornea diameter, anterior and posterior segment depths, and lens thickness and diameter as well. So all measurements were analyzed using one-way ANOVA and post-hoc Tukey-Kramer tests to evaluate any significant difference. You can see at the poster that eyes from 6-month-old nano pigs and 6-month-old mini pigs, they don't differ as much in when looking just the structure. So in our results, what we found is that no significant differences were observed in axial globe length, lens width and depth, anterior segment depth, and subocular tissue weight between any strain. Although minimal, statistically significant differences were observed in transverse globe length, which eyes from Yucatan mini pigs present the largest size, but the difference is still minimal, I would say. And corneal diameter had a little bit of a difference as well, where the Yucatan pigs had also a larger corneal diameter. And posterior segment depth, which is the same. The Yucatan mini pigs present the largest anterior posterior segment depth. So in the table on the poster, you can see all the measurements and the standard deviation. And we concluded that while there is some statistically significant differences in some of the globe measurements among the three species, the eye measurements from all three species are comparable in weight and size and equally suitable for use in ocular studies. So the use of a nanopig doesn't differ as much with the use of Yucatan pigs The only difference is in body size, but the eye present almost the same measurements. So I would like to acknowledge all the staff at Sinclair Bioresources for the collaboration in shipping us those samples. Thank you so much.